And it's my great pleasure to introduce our speaker today, who is a member of our network from the very beginning and very valuable member, Professor Lars Lewejohann from the German Center for the Protection of Laboratory Animals and Freie Universität Berlin. And Lars has studied biology and philosophy at the University of Münster, and already for his diploma thesis back in 1999, he studied and evaluated the housing conditions of laboratory mice from the animal's point of view by using the preference tests then. Then in 2004, he completed PhD project that was evaluating a test battery for murine models used in biomedical research. After working in Münster, he had 2 professorships for behavioral biology at the universities of Osnabrück and Göttingen. And since 2017, he is a professor for animal welfare and refinement at the Freie Universität Berlin and the head of unit lab animal science at German Center for the Protection of Lab Animals. And in his lab and his own interests, they are working on new concepts of social, cognitive, and environmental enrichment that are developed in order to counteract animal boredom. Also, individual differences, the interplay of cognition and emotion and animals' point of view with regard to better housing conditions and experimental design are among the current research topics. So we will be very excited to hear Lars talk on improving laboratory housing systems for the benefit of animals and for sake of better science. Please, Lars, the floor is yours. Screen.
Thank you so much, Vootele. And yes, thanks all for coming. And I heard that a lot of people are here, and I'm, well, flattered. Thank you very much for your interest. And I hope I do not preach to the converted here, but this should be a plea for improving laboratory housing systems for the benefit of mice, but also for the benefit of better science. So just to jump right in. Let's see if this works. You should see the second slide now. These are current numbers from animals used in Europe. So overall in Europe, 8.5 million animals were used as experiment or used in animals related to the experiments. There were also 10.4 million additional animals. So these were animals for tissue and organ samplings, but also for breeding and maintenance, and the so-called surplus animals. So animals that are bred for the purpose of being used in experiments or having the wrong genotype, the wrong sex. So animals that were actually not tested, but still these are also animals that are of concern if we are talking about animal welfare and about improving housing conditions. So most of these animals are mice, basically all over the world in experiments. This in 2022, the statistics only 48% of the animals used were mice. This usually sums up to around 60%. In that year, there were an exceptional high number of fish being used. And I looked this up, and this is probably related to a very big experiment done in Norway, where they tested salmon lees, or treatments for salmon lees. So usually more than 48% of the animals used are mice. And if we look at mice, so this is a picture from a standard facility I took this morning, actually. So this is where mice are usually housed in, and the other picture on the left side I took from the Techniplast website. So this is one of the most commonly used cage systems. It has a floor space of 535 square millimeters. And according to the EU legislations, this holds space, depending on the weight of the mice, for 5 to 8 mice in such cages. And this might be problematic. Mice spend most of their lives in such cages and not in the experiments by itself. So in this time scale, I took or put together two figures from publications. One is the development of out through old age of the mice, and the other one is influences that will actually affect behavioral outcomes used in behavioral tests. So exemplified here as these short tests, some of you might already know. So tests on anxiety-related behavior like the elevated plus maze, but also tests on learning and memory like the water maze here depicted. What happens before these experiments can affect the outcomes of the experiment and diverge into different directions. And this is one of the reasons people in recent years thought that it might be a good idea to have a high amount of standardization and hygienic concerns to try to rule out all these confounding factors that might affect the results to well, not a really great success actually, because there still is a huge amount of variability in the data, and we might look into this later why this is so. But the message here is that the animals are kept in these cages I just showed you, and being in such a restricted environment, in our view, is related to boredom, because there's not much you can do all day. So we took a look into the literature and looked up parameters that are related to animal boredom, which is not much that is found in the literature, but what we found here on the right side, parameters connected with boredom, is, for example, drug-seeking behavior, depressive-like behavior, also hair pulling, the way time is perceived, novelty-seeking, risk proneness, and behaviors like being inactive but awake. I will show a figure later on. And we found out that there's a huge overlap with parameters that are connected with environmental enrichment, which is improving the housing conditions of the animals by adding something that the animals can interact with actually, and this might also be one of the main treatments that we can perceive for treating boredom. So there's a lot of overlap between these parameters that are connected or affected by environmental enriching the home cages of the animals, including social behavior, affective well-being, aggressive behavior, but also circadian rhythm or abnormal behavior, for example, stereotypic behavior. So with regard to stereotypic behavior, I would like to show you a video. On the right side, you see a so-called standard cage, which is a cage with not much environmental enrichment in it, except for a house where the animals can build their nests, nesting material, bedding, the food hopper here, and there's a water bottle somewhere. On the left side, you see an enriched cage with a second plane here, some 3D printed elements I will introduce later on, something to gnaw on, nesting material, and a running disc. And on the left side here, you see this particular mouse performing repetitive behavior, so stereotypic behavior, like circling or jumping on the guitar, on the cage lid. Also, gnawing on the cage lid is something that is typically seen as stereotypic behavior. So repetitive elements that are usually being showed more in enriched in standard or in conventional cages compared to environmentally enriched cages. Another symptom of boredom I mentioned, and I took this from a publication of Georgia Mason's group, is inactive but awake, something that you might notice if you know the definition of that sort of behavior. So you see a mouse standing still, the eyes are open, but it's actually doing nothing. So it is obviously awake, but it is inactive and just standing there. And you see that there are huge differences between mice from different strains, C57 Black 6, Bulb C, and DBA strain, but also between different housing conditions. And you see this in the C57 Black 6 here, that mice from the enriched housing conditions show less of this inactive but awake behavior compared to animals from the non-enriched conventional housing systems. Another symptom that is related to boredom, at least in humans, is a state called trichotillomania, conventionally called barbering behavior, so hair pulling. And this is in human psychology related to the state of boredom, and we suggest that this is also related to boredom in laboratory animals. We did a short survey in German facilities, and we found that all of the facilities that responded have observed this type of behavior, so the hair pulling behavior, in well, round about a little less than 10% of the animals that were affected by this. So this is something that should be of concern, especially as the mice tend to also not only pull the fur but also the whiskers, which brings to concern a lot of things that probably be affected by not having whiskers in behavioral tests and not being able to use this type of stimuli. Here's another thing that we would relate to animal boredom. That is a phenomenon called contra-freeloading. Contra-freeloading is by definition a behavior when an animal is working for getting access to a good which it also can receive for free. So in this experiment that we did here, the home cage of the mice was connected to a cage where they had to work for getting access to different types of liquids. And as a control condition in this experiment, we provided water for free. So only one nose poke had to be done in an IntelliCage system for getting access to water. And in the working corner, there was usually different liquids, but as a control conditions, also just a bottle of water where the animals had to do increasing numbers of nose pokes up to, so this was increased by 1 nose poke required per day, so up to 7 nose pokes were done for getting access to water while it was available for free. So they tend to work for something which they also can get for free, so they have too much time on their hands or paws and just continue working, although they can get it for free. This is a phenomenon observed in many different animal species, actually, that they tend to work for something that they also can get for free. Interestingly, I found in the literature that cats don't do this. So if they get something for free, they don't work for it anymore. So how to treat boredom? And I would like to explain this a little bit by this figure I compiled without data, but just based on the literature. So what we suggest here is that we have on the x-axis the amount of environmental challenge that is presented to an animal, and on the y-axis here the welfare state, with the higher values here being better welfare. And if you are in a severe stressed condition, so under distress. This is too much environmental challenge for the organism to deal with. So this will probably be related to poor animal welfare. But in our view, the same is true if there's an understimulation. So if there's no challenge at all, if you're living in a boring home cage, then you are in a state of being understimulated, and this is also related to bad animal welfare. So the ideal would be probably something in between, to have environmental challenges to a certain amount, something to do and something to cope with, and this right amount of stimulation probably leads to an increase in animal welfare. What we did as a reference system for analyzing animal behavior in mice in a state where we believe that these mice are probably not bored is such a semi-naturalistic housing condition, where we had a three-dimensional space used by the animals here, connected by these tunnels. All animals had RFID transponders in their neck region, so we could monitor 24/7 the activity patterns, the way they use the space. So if they are just roaming around at one of these levels and have many antenna contacts here, or if they roam around in the whole semi-naturalistic enclosure, and thereby also have a high number of antenna contacts, but a high number of different antenna contacts, which is called roaming entropy. So the more space the animals use, the higher the amount of this roaming entropy is. And this is a reference system for the boredom project here, where Paul sat in front of the cage, did manual observations of behavior, and we measured, aside from the automatic part here, a lot of other things, including different behavioral patterns. First of all, one thing that is often named a concern with regard to environmental enriched conditions, and probably some of you might think that this is also a concern of such a semi-naturalistic enclosure, is the idea that this might bring about more variability in the data. And I would like to point out here from physiological parameters that were also measured with regard to bone health, so different parameters of bone density, of cortical thickness of the bones, of the length, and so forth. And we also analyzed how the variability was affected in different housing conditions, including standard housing conditions, enriched housing conditions, and the semi-naturalistic housing conditions. And we did not at all observe a systematic increase of the physiological parameters being more variable in relation to the different housing conditions. So there was no such increase in variability that is often named a concern. And this is actually in line with actual literature data from other studies that also observe no such increase in variability related to environmental enrichment. So basically this concern can be busted here. As I already said, in the day-to-day behavior, Paul observed no stereotypic behavior at all in the semi-naturalistic enclosure, and interestingly, play behavior up to a very old age of 18 months of these animals. They still show play behavior in the semi-naturalistic conditions, which is something that we have not found in the literature in standard housing conditions. So play behavior in standard housing conditions usually is shown only by very young animals and very seldomly at older age of about 60 days and not at all in aged animals. But in the semi-naturalistic housing conditions, these mice still continue to show such play behavior. In the introduction, Vootele already said that in my diploma thesis, I already started analyzing different housing conditions for laboratory mice. So in 1999, this was considered a standard cage for mice, and this was considered an enriched cage. So basically same-sized cage with a shelter in there, a climbing frame, and not shown here some nesting material. That was the enriched conditions. Nowadays, the enriched conditions from 1999 in the study of 2020 is basically reflected as a standard condition. So there's also a shelter here, nesting material, and some wooden blocks for gnawing. And now the enriched conditions comprises a second plane here, elements that they can, so this is a paper hut and a running disc in here. So enriched conditions nowadays are much more enriched than they were 25 years ago. Here's some enrichment elements that we use in our lab. For example, structural elements like different types of housing of these shelter conditions, including these paper huts that they can destroy over 1 or 2 weeks and use as additional nesting material. Structural elements that can be attached to the cage lid. These plastic things where, for example, cardboard tubes or plastic tubes can be attached to the cage lid you see here. These second planes that can be added here, or wooden structures to structure the home cage of the animals. This is a category of nesting and shelter materials, different bedding material, and also can be used for nesting, like these nest pads here, or these cotton rolls and paper tissues. And the category of activity and cognitive stimulation includes, for example, these running discs that we provide our animals with, that have some advantages over the conventional running discs often being used in the cage. Especially the mice don't have to bend their backs as they have to in the running wheels. If they're using these running discs, they can more or less be in a natural position on this running disc and increase their activity. These are different examples from we use our 3D printer for providing such riddles where millet seed, for example, can be provided, and the animals can open these doors here to get access and work together on these things to get a food reward. This grid ball is attached to the cage lid by this chain here, so it moves around when the animals try to remove the nesting material that we stuff into this grid ball, so they can interact quite a long time with this structure to pull out the nesting material. This is a tube that is blocked with stones, something that mice are very keen on removing the stones out of such tubes if they are blocked. This lasts only for a few minutes, and then usually all the stones are removed. And this is also something that we 3D printed. This is a ball with a hole in there. We can fill the ball. So this is two parts that are screwed together, and we can fill it with millet seeds, and if the mice move it around, eventually millet seeds drop out and they can interact with this ball in order to receive food rewards. In her PhD study, Ute Hobby-Siefken analyzed the behavior and how they interact with these animals, with these elements of enrichment in their housing conditions. And Ute observed that the behavior I showed you before, inactive but awake, was significantly increased in conventional housing conditions compared to the enriched housing conditions. And also the stereotypic behavior was significantly reduced in the enriched housing conditions, but still in this sort of enriched housing conditions compared to the semi-naturalistic environment where no stereotypic behavior was ever observed, we still saw this stereotypic behavior in the enriched conditions, but significantly less compared to the standard or conventional housing systems. Ute also analyzed the percentage of usage or the way these elements that I just presented were used. Interestingly, the running discs here were used to a high amount of around 30 to almost 50% of the time, depending on the other elements that are presented. And she also categorized if these elements were used actively or inactively. For example, these housing structures, house ball was used more often passively, so they built the nest under this structure, while the running discs were used more actively. This is from a systematic review where we scanned the literature for effects of enrichment. And one caveat that is in this study I already mentioned. So what was considered environmental enrichment in 1999 is different from what is considered environmental enrichment today. But so we used environmental enrichment here as an umbrella term and scanned the literature. But the control group also always reflects the conventional housing systems, while the enriched category here reflects an environment that is at least more or providing more elements and more structure than the conventional one. The general effect, or first of all, how to read this figure here, the thickness of the bars or the height of the bars here represents the number of studies where such effects were seen. And if this is shifted to the right, then there's an increase. If it's shifted to the left, there's a decrease. And if there are no differences found between the different housing conditions, then it's depicted here in purple as neutral. So generally speaking, there's an increase in or an increased effect on cognition of environmental enrichment. So the performance in cognitive tests is improved due to environmental enrichment. The same applies for motor function and affective well-being. Social behavior also usually is found to be increased, and exploratory behavior as well. There is no clear effect on aggressive behavior and on activity, but there's a decrease in abnormal behavior. And we also scanned the literature for effects on glucocorticoid stress hormones in mice. And there's also not a clear picture. Sometimes it's increased, sometimes it's decreased. Most of the time, there's no differences found between the different housing conditions. We differentiated in the systematic review between rats and mice, but basically the same effects are shown for rats and mice. And what we see here is the effects of the different sex used. So what we found in the literature is a bias that is still present in most of biomedical research, with being a bias towards males being used in such studies and less females. In those studies that uses both sexes, also less than the whole number of studies where only males are used. Interestingly, the effects are basically the same, going in the same direction. And what I would like to point out here is the effect of aggressive behavior, which one would expect to be increased in male mice, showing more aggressive behavior than female mice. But also here, there's no clear picture that environmental enrichment will always increase aggressive behavior or decrease aggressive behavior. This certainly depends on the type of environmental enrichment being used. But no such clear effect that that one of the concerns that is often mentioned in the literature, that environmental enrichment will increase aggressive behavior, we could not find such an effect in the general tendency. Is the effect of different time of application or length of application of environmental enrichment. So short is only a few days, mid is a few weeks, and long is basically for months. And we see a slight increase. The longer the better, or the longer the more effects on these types of behavior. But also already a short introduction to environmental enrichment, or introducing environmental enrichment for a short time, also already shows clear positive effects here. So there are effects on the behavior, but let's ask the mice themselves what they think of it. I did this in my diploma thesis, so I asked the mice if they would rather live or prefer to choose, if in a choice test, living in an enriched compartment compared to the standard housing. They all choose the enriched compartment, and in this study we now analyzed individual behavior of mice that are still living in a group situation by using RFID transponders implanted in the neck region of the animals, and introducing this mouse position surveillance system, where we connected the cages by a tunnel and two RFID antennas, and we now can evaluate the position of the animals in these systems and measure the time they spend with different types of enrichment that we present in the different cages. And although they are still living in a group situation, we can monitor individual behavior within the group using this system. This actually took a long time until we managed to have this system, because animals pass through these tunnels so quickly that sometimes the RFID antennas did not read them. So Anne Jaap spent a lot time figuring out how to slow the animals down, and a technician, Birk Ormersbach, developed new RFID readers that have a higher sensitivity for the RFID antennas, and including a software system that we now have, we can now claim that we see the animals 100% of the time, the exact position of the animals. So this system is working, and it's freely available. If you follow this publication, you can download it, and also can get the new RFID readers from us if you want to. So we have everything published as open access and open source. In her PhD study, Ute then analyzed how much time they spend with the different animals. So from the different categories of environmental enrichment elements, she did binary choice tests, granting them access. For example, in one cage was this grid ball, in the other cage was this 3D printed thing with millet seeds in there, and then she measured the time the animals spent in these different cages, and we had a combination of many of these binary choice tests, and thereby could derive rank order which of these elements is perceived as being or is more important by measured by spending more time with it. So the winner for the activity introducing elements here was a grid ball, while the sliding puzzle, this one here, was a least preferred. What you see here is a measure for how the animals agree on this decision. So the higher this number is, the less agreement was in the preference for this. So while there was more or less clear preference for the lattice ball, it was not so clear the difference between the elements on the lower range here. This was even more the case by comparing different housing systems that we provided. So in the active time, so in the nighttime when the mice were awake, they moved around in both cages, and one might see a small preference for the paper house. But in the inactive time when the animals were sleeping, basically all of these different types of housing were suitable for building the nests in there, and there was no clear preference for one type of these housings. I now would like to present you studies from our research cluster Science of Intelligence, where we analyze intelligent behavior of mice, so how they solve such log boxes. But these elements can also be used as a sort of a cognitive enrichment, where the animals engage with these apparatus in order to get a reward. And we observed that the animals actually queue up for getting access to the cages where we provide these log boxes. So these are comprised of different locking mechanisms. The mice has to open in the right order to get a food reward. So you saw that this lever was moved. Now the stick is pulled out. The stick is blocking this ball here. No. Now the ball is removed, and the ball was blocking this sliding door. Now the sliding door is moved, and the mouse is receiving the food reward, which is in half of an oat flake here. We use these systems to have an analysis pipeline using 3 cameras from different angles, from a top view, from 2 side views, and then reconstruct 3D representations of different body parts of the mice using deep lab cut, and then doing a pipeline of how much time the animals interact with the different animals. So this is not the same mouse, I guess, but this is exemplifying how this analysis pipeline is working. So the ear and the nose position is tracked here, and every time the colors blink here, the mouse is interacting with this type of logbox detected automatically by the analysis pipeline, and this was evaluated by human annotators, and these are the matrices showing that we have a quite high correlation between what the human annotators observe and what the automatic system is observing. Interestingly, after only a few trials, actually all animals were able to solve the puzzle. Not in all trials all animals performed, but on an individual level, all of them eventually solved the lockbox and received the reward. And with regard to the analysis of intelligent behavior, we are still a little bit stuck. So we are trying to make this more complicated because they were actually too fast to learn this, and we have systematic difficulties to differentiate between the motor learning part and the part of what we actually would like to see, that is an understanding of how the mechanisms interact. And so we are still on our way trying to figuring this out. But this was for the purpose of this talk, exemplifying that this can be used as a cognitive stimulation for the animals, and that such experiments can actually be done in the home cage of the animals by using different camera views and opening a door to an adjacent cage and let the animals into that new compartment and solve such tasks. Other examples of home cage-based testing are shown here. So this would be the home cage, home environment of the animals. This is an animal gate where animals also with an RFID antenna can enter this cage individually and then work to get access to this cage. And this is a case study where we try to measure the motivation for getting access to a cage with much bedding where they can dig in. And this is how it looks after one night. So indeed, the animals get access to this cage and dig in there and build burrows and so forth. This is a new system that is established in our lab for analyzing behavior in natural conditions, and I termed this happy mice. This is just right after the animals were introduced into the system. So this is a modular semi-naturalistic environment where we have 6 of these modules connected by tubes with RFID antennas, and the mice can now get access, roam around in these cages where food and water is provided. One of the elements is a so-called performance platform where we can introduce tests to the mice, but the other compartments are always nesting material and food and water. And one idea that we are trying to use such systems for is that we still lack a lot of measures for assessing how, for example, the severity of certain procedures or the welfare state of the animals if we just use short tests in open field tests, for example, or light dark boxes or even scoring systems where this has to be done manually. And the idea now is that using such systems and having cameras on each of the modules and also microphones in there for observing them 24/7 will fill a lot of the gaps for actually what we like to know about our experimental animals in systems that allow more natural behavior than the standard or conventional housing systems most laboratories use. So this is a short example that Anna Klenova did in this new environment, analyzing ultrasonic vocalization. So you see the top view of the camera and two animals interacting here. You did not hear this because I did not transfer the ultrasonic vocalization, but you see here the plots where you see certain types of vocalization being emitted by these two animals interacting. And if you see a different type of interaction here by these two animals, you see that the vocalization actually is a lot different from the sociopositive interaction. And Anna spent a lot of time manually analyzing these ultrasonic vocalizations and found well, very interesting differences that probably are or can be used for automatic detections in such systems in the hopefully near future to detect different types of social interactions by using bioacoustics. Okay, coming back to the introduction where I showed the number of animals used in Europe and the animals that are not used in the experiments, I would like to present this table here for here are the experimental animals, and there might be concerns how we can improve the live testing and housing conditions of these animals, but there probably are less concerns for improving the conditions of the animals after the experiment or for animals that are actually not really used in an experiment. So there's a lot of room for improvement here. One thing that one can do is, for example, introducing running discs after the experiment. These animals are also around about 18 months of age, and after the experiment, Pia provided these running discs, and you see here's an old lady for the first time on a running disc, and in the second video, the same animals 2 weeks later have learned to use the running discs and even run on them in pairs or even more. And so the take-home message here would be it's never too late for environmental enrichment. With that, I'm at the end of my talk and happy to take questions.